All right, in this, our second screencast of Chapter 7, we're going to continue on with our discussion of the composition of the plasma membrane. Plasma membrane continued. Pardon me. So, membrane proteins, we were talking about the composition of the membrane. We talked a lot about the lipids. We talked about fluidity, the importance of maintaining fluidity, a little bit about the evolution of the lipids in the plasma membrane. So now we're going to turn and look at the other components of the plasma membrane that make it the mosaic that it is. And let's look at the proteins. So a membrane is a collage or a mosaic. If you think about like what a mosaic is, right? You think about those, a mosaic floor or a mosaic mural, and it's a bunch of little tiles, like tiny little squares of different things put together to make kind of a big picture. And that's what the plasma membrane is like. Lots of different things put together to make one large structure. So there are a lot of different proteins. Sometimes they're grouped together, sometimes they're not. And the proteins determine most of the membrane's functions. So the phospholipids give it structure, give it its ability to become a barrier, and then the proteins determine the function. And you have a couple of different kinds of proteins. Peripheral proteins are bound to the surface of the membrane. So if I have my lipid bilayer here, a peripheral protein would be kind of on the surface, either inside or outside. An integral protein penetrates the hydrophobic core. So it'll either completely span the core, or it will kind of be not go all the way through. So excuse me. We call these integral proteins. Okay. If the proteins span the entire, like this one here, the membrane, they're called transmembrane proteins across the two sides of the proteins. Okay. And again, these proteins that integral proteins must be amphipathic because they have to have both hydrophobic and hydrophilic regions to be in both parts of the membrane. Okay, the hydrophobic regions that are internal to the membrane bilayer generally have stretches of nonpolar amino acids, often coiled up into alpha helices. And here, if you look, so this area of the protein would be hydrophilic. I'm just going to write philic. Okay, this area in here, hydrophobic. Okay, and so a lot of these alpha helices, okay, will be stretches of nonpolar amino acids, so they can live in the nonpolar region of the plasma membrane, and then this area would be polar and hydrophilic. So 6 major functions of membrane proteins. And again, remember, proteins are by far our most diverse group of macromolecules. So they function in transport, enzymatic activity, signal transduction, that's chapter 11, which we're going to do next, cell-cell recognition, intracellular joining cells together. Cell-cell recognition helps cells talk to each other so they know what's going on, and attachment to the cytoskeleton and extracellular matrix to hold our cells in the right shape and in the right place. So here's an example of this. So transport, show some ions getting through the plasma membrane via a channel protein or active transport. Enzymatic activity, enzymes can embed themselves in the membrane. We're going to see a lot of this when we talk about cellular respiration and photosynthesis, because membranes are very powerful things. And as we go through the rest of this chapter and then talk about them again in chapter 9 and chapter 10 with cellular respiration and photosynthesis, membranes are very powerful. They are super fancy, very cool, and they help us to do a lot of things. Signal transduction helps get messages in and out of cells, tells cells what to do and how to respond to certain situations. Cell recognition. This helps cells talk to each other and communicate and help cells find other cells. A lot of the carbohydrates are involved in cell recognition. You'll see this is a glycoprotein, which means it has a carbohydrate. Intracellular joining. These are like plasmodesmata, tight junctions, gap junctions that help cells stick together and not come flying apart, which is good for like your organelles and stuff. Or not your organelles, your organs. There we go. And attachment to the cytoskeleton. Cytoskeleton helps hold cells in place. And the reverse. Cytoskeleton helps maintain the interior structure of the cell, and extracellular matrix out here helps to keep the cells in place. So carbohydrates, again, as I just mentioned in the previous slide, they have to do with cell recognition. So the carbohydrates kind of act as like signposts, almost like name tags on the outside of the cell. So cells recognize each other by binding to surface molecules, almost always containing carbohydrates on the extracellular surface of the plasma membrane. Okay, carbohydrates can be attached to lipids, and then that would be a glycolipid. Glyco means carb. Okay, or more commonly to proteins, and then that you get glycoproteins. Again, the glyco meaning the carbohydrate part. Okay. Carbohydrates on the external side of the plasma membrane vary among species, individuals, and even cell types in an individual. And again, because they help recognize a specific type of cell and a specific type of organism. And here, and we'll talk about HIV much more when we talk about viruses, but HIV infects your cells in an incredibly unique way in that it has a plasma. So this is the virus particle in here, and it has a plasma membrane capsule around it that has glycoproteins on it. And these glycoproteins basically are little tags that recognize your T cells. Okay. And so basically the HIV, this capsule takes the HIV to the exactly right cells. They bind to receptors on the cell surface, these guys right here. And the receptors don't know what's inside of this capsule with the glycolipid, but it knows that the glycolipids are what is supposed to be in their cell. So it actually brings the HIV particle inside the cell. And it's really quite diabolical. And we can talk about that. We'll talk about that more when we talk about the virus chapter. But again, this is, most people have both of these receptors. Every once in a while, you'll get somebody who doesn't have the CCR5 receptor on the surface of their T cells, and they actually can't be infected with HIV. So they're immune to it, which is really kind of cool. But it all has to do with these glycolipids, these proteins that help you recognize the cells recognize each other and cells communicate. Okay, so let's talk about where and how membranes are made, and also the sidedness of membranes, inside versus outside. And this has a lot to do with the endomembrane system that we learned about in chapter 6. Endomembrane system. Okay. So membranes have distinct inside and outside faces. So one surface will always end up being the outside of the cell, and one side will always end up being the inside of the cell. So, and it has to do with asymmetrical distribution of proteins, lipids, and those carbohydrates in the plasma membrane. And then when the membrane is built by the ER and by the Golgi, it has to be made in just the right way. And if you look if you want something to end up outside of the cell, say these proteins are going to end up outside of the cell. In the rough ER here, and in the Golgi here, we have to make sure, actually, that if we want something to be outside the cell, it has to be made inside of a vesicle. Okay, and if you look here, if we make it inside of the vesicle on the ER, it's going to be inside of a transport vesicle as it's going over to the Golgi. Then when it fuses to the Golgi, that'll put it on the inside of the Golgi, and then it gets packaged on through, and it buds off into another vesicle. It's on the inside of the vesicle still, but as the vesicle fuses with the membrane, the things that are inside the vesicle end up being on the outside of the cell. Okay, so if you want something to end up on the outside of the cell, to be secreted or be on the membrane, it needs to be made interior to the endomembrane system. And we'll go over this. It's sometimes tough for people to get this concept. Okay. All right. So let's move on to a little bit more about the selective permeability of the membrane. So the membrane structure itself, just the lipid bilayer, lends a lot to the selective permeability. So cells have to exchange materials. It needs to bring in food, nutrients, water, things like that. It needs to get out waste that will become toxic. Okay, so plasma membranes are selectively permeable. Some things get in and out, some things don't. And this helps to regulate molecular traffic across the membrane. So anything that is hydrophobic and small can dissolve straight through the lipid bilayer and pass right into the cell. Okay, so anything that is nonpolar, like hydrocarbons, okay, or oxygen. Oxygen molecule is nonpolar. It can zip right through. Polar molecules, like sugars or large proteins, do not cross the membrane easily. So if we want them to get in, which a lot of times we do, they need help. And things get helped across the membrane by transport proteins. And so what transport proteins can do is they can make like a little safe channel for hydrophilic substances to cross the membrane. These are called, oddly enough, channel proteins. So they have a hydrophilic channel through the center of the protein that that ions or other hydrophilic substances can use as a tunnel through the membrane to get into the cell. They're also special because we need so much water, and water is, of course, polar. There are special channel proteins called aquaporins that get the water into the cell quickly as possible. Okay. Other types of transport proteins called carrier proteins. We're going to learn more about these in a future screencast. They bind to molecules and change shape to shuttle so it's not like kind of an open tube, like a tunnel, like the Lincoln Tunnel or something like that, but it's a little bit more selective, and it only allows very specific molecules through. Okay, so transport proteins are specific. Channel proteins, if you fit through, you can go in. Transport proteins are more specific. Okay, and that's it for this part. And we're going to move on to transport in the next screencast.